discussing the tools of molecular biology. Now you may ask yourself, why do we have to learn about the tools of molecular biology? The simple answer is because they tested on USMLE. And so we're going to talk about the most common molecular biology tools that we use in order to study DNA and proteins. The other reason to learn this is that a lot of these tools have been adapted for diagnostic and therapeutic uses, and so it's important for physicians to understand the basics of these techniques so they can understand those processes. In this lecture, we're going to discuss the following things. We're going to talk about how we analyze proteins, both how proteins are separated from one another and also how they're detected. We're also going to discuss how we analyze DNA using restriction endonucleases, southern and northern blotting, PCR, and recombinant DNA technology. First of all, let's talk about protein analysis. In order to study proteins, you have to be able to take a tissue sample or something that has a lot of different proteins in it and separate those proteins one from another. Now you can do that, you can separate proteins from complex mixtures using what we call differential separation techniques. These are techniques which separate proteins based on their physical properties, based on things like size or charge or their ability to bind some other substance. An example of how we separate proteins by size is in gel filtration. In gel filtration, we have a column which contains a whole bunch of porous gel particles. And each of these little particles, is shown in the schematic to the right, has little pores in it. Now, the proteins that are very small can enter the pores in these particles, and thus they get trapped and they run through the column at a slower rate. The larger proteins don't enter the pores, and they run through the column at a faster rate. What we do then is we sequentially collect fractions of material that flow through these columns, and each sequential fraction contains smaller and smaller proteins. Another method that you can use to separate proteins by size is what we call sodium dodecyl sulfate polyacrylamide gel electrophoresis, or SDS-PAGE for short. In this, we take a protein mixture and we mix it with a detergent called sodium dodecyl sulfate. This denatures the proteins, and it imparts a negative charge to them, so that the proteins are very negatively charged. You take this mixture then and place it at one end of a gelatinous matrix and apply an electrical field. The proteins will run through the gel towards the positive charge because of the negatively charged SDS which is attached to them. The speed at which they run through that gel is determined by their size. So larger proteins will run through the gel at a slower pace, while smaller proteins will run through the gel at a faster rate. And so you can separate proteins by size according to how far they run through the gel. That's illustrated in this following schematic. Here we've illustrated a gel. We load a protein mixture into a well in that gel, and we apply an electric field. The electric field is going to be negative at the origin of the gel and positive at the far end of the gel. So proteins loaded into that well then migrate, according to their size, towards the positive charge. The proteins at the bottom of the gel will be smaller. The proteins that end up at the top of the gel will be larger. One of the applications of electrophoresis is in the diagnosis of diseases. You'll recall from earlier discussions about sickle cell disease that this particular disease is caused by a defective form of hemoglobin called hemoglobin S. Hemoglobin S has the property of moving at a slower rate through an electrophoretic gel than the normal hemoglobin A does. And so the results of hemoglobin electrophoresis are shown below. In hemoglobin electrophoresis, you'll see somebody that is normal, has no hemoglobin S at all, will have a single band that migrates at the hemoglobin A position. Somebody who has hemoglobin S, all of their hemoglobin will migrate at a much slower rate, such that it ends up in a different position than hemoglobin A. Somebody who has sickle cell trait, that is someone who is heterozygous for both hemoglobin S and hemoglobin A, will produce both of the proteins, and they will run in the pattern shown below. That was a diagram. A real electrophoretic gel, hemoglobin electrophoresis, is shown in this picture, and you can see that there are several individuals which appear to be heterozygous for the disease because they have two bands, some that have hemoglobin A alone. These are normal individuals. Now, in addition to separating proteins by size, we can also separate them by charge. One of the ways that we do that is using ion exchange chromatography. Ion exchange chromatography is similar to gel filtration in that we run the protein mixture through a column. But instead of using the porous gelatinous beads like we did in gel filtration, instead we run the mixture through a column that contains negatively charged beads. Now, negatively charged proteins will be repelled by these negatively charged beads, and they flow through the column very quickly, while the positively charged proteins will bind to the beads. So we've been able to separate negatively charged proteins then from positively charged proteins. Now, the positively charged proteins that are stuck to the column have to be released somehow. And we do that by running a concentrated salt solution through the column. The sodium ions in the salt solution compete for binding of the beads with these positively charged proteins. And as we add increasing concentrations of that salt solution, we sequentially release more positive proteins. So the proteins released at low salt concentration are not very positive, while those released at higher salt concentrations are more positive. They have more positive charge. And so as we collect sequential fractions, then we get protein mixtures that have proteins consisting of more and more positive charge. Another way that we can separate proteins by charge is similar to electrophoresis. It's a specific type of electrophoresis called isoelectric focusing. Now, every protein has a particular property that we call its isoelectric point. This is the pH at which the protein has a net charge of 0. Now, proteins that are positive will have a basic pi. That is, they will only have a net charge of 0 at pHs greater than 7.0. Proteins that have a net negative charge will have an acidic pi. That is, they will only have a net charge of 0 at pHs lower than 7.0. In an isoelectric focusing gel, you run proteins through a gel that's very similar to an SDS page gel, except there are two differences. One, we don't use SDS, so the proteins have their native charge. They haven't acquired all the negative charges that we get when we mix it with SDS. The other difference is that the gel we run them on has a pH gradient. It's acidic at one end, and it's basic at the other end. So we put the proteins on the gel, we apply a charge, and the proteins will run in the gel until they reach a point in which the gel pH is equal to that protein's isoelectric point. At that point, the protein now has no charge, and so it stops moving in the electric field. And that way we're able to separate proteins based on their isoelectric point. Once we've separated these proteins, then we have to be able to detect specific proteins. We can detect specific proteins using antibodies, which have been produced by injecting an animal with the protein and having that animal raise specific antibodies against it. One technique for doing this is what we call enzyme-linked immunosorbent assay, or ELISA. In ELISA, what we do is we coat the bottom of a dish with antibody that's against whatever protein we're looking for. We can then take a protein mixture, add it to the dish, and allow the specific protein that we're interested in to bind to the antibody. Once that's bound, we can then wash off the other antigens that don't bind the antibody, and then we can add a second antibody. The second antibody has an enzyme on it that produces a fluorescent product. So the amount of the protein that we're interested in that dish is measured by the intensity of the fluorescence produced by that secondary antibody. This is illustrated schematically on this slide. Here we have our dish, which is bound to antibodies specific to a particular protein. We add a protein mixture. You can see there are red, blue, gray, black proteins of different sizes and shapes. The white proteins bind to this specific antibody to them, and the remaining proteins are washed off. We then add the secondary antibody that's linked to an enzyme. We add the fluorescent substrate to that enzyme, and the secondary enzyme then glows. And the intensity of that fluorescence then is reflective of the amount of protein that we have in that dish. Now, another way that we can detect proteins using specific antibodies is Western blotting. In this, we put a protein mixture on an SDS page gel, we separate the proteins by size, and then we transfer the proteins to a nitrocellulose membrane. This membrane is then incubated with enzyme-linked specific antibodies, similar to what we saw in ELISA. Then, once we've done that, then the enzyme substrate is provided, and the antibody-protein complex is detected on X-ray film by radiography. This is shown schematically here. Here the proteins in step 1 are separated by size. In step 2, they're transferred to the nitrocellulose membrane. In step 3, the enzyme-linked specific antibody is added, and creates fluorescence. In step 4, the membrane is exposed to X-ray film, and the specific antibody appears as a band, a specific single band, which appears on that X-ray film. So these are different ways that we can separate and detect and work with proteins. Next, we're going to talk about techniques that we use to analyze DNA. Now, in order to work with DNA, you have to first be able to reduce DNA to a reasonable size. Remember, we talked about before that DNA within cells is in chromosomes, which are millions and billions of base pairs long. And that's not workable. So in order to work with DNA, we've got to cut it up into manageable sizes, manageable pieces. And we do this using specific enzymes, which are called restriction endonucleases. These are enzymes which are derived from bacteria, which cleave DNA at specific sites. Now, restriction enzymes always cleave DNA at the same site. And these sites are usually what we call palindromes. These are sequences that are read the same, 5' to 3' on either strand. An example is shown below. Here, one strand reads from 5' to 3', GAT, ATC. And on the other strand from 5' it reads GAT, ATC. But this is the reverse complement of the other strand. And so this is what we call a palindromic sequence. Now, restriction endonucleases can cleave DNA either bluntly, that is, leaving no overhang, or they can cut in a staggered fashion, as shown below here, leaving a non-base paired overhang. Now, we can do with DNA just like we do with protein. We can separate the DNA fragments by gel electrophoresis, much the same as we do for proteins. We add the DNA mixture to a gel, we apply an electric field, and the DNA runs towards the positive pole. Remember, the DNA is negatively charged because of the phosphate group. Then we have to be able to detect these DNA fragments, similar to what we do in proteins. So we use a very similar technique, except this technique is called southern blotting. So we run the fragments out on a gel, we transfer them to a membrane, and then we detect them using a radio-labeled complementary DNA probe. That is a probe that has radioactive tag on it. Then we can expose that to X-ray film, and we can detect a band that represents that specific sequence. Now, that's southern blotting. Northern blotting is exactly the same, except for rather than using DNA, we're using RNA, and again, detecting it by a radio-labeled complementary DNA probe. Now, we can use this combination of restriction endonucleases and southern blotting to do what we call genotyping of particular individuals. Now, the restriction endonuclease cleavage pattern is different for different alleles in a population, and consequently, all of our DNA, all of our chromosomes have different restriction endonuclease cleavage patterns. That is, the cleavage pattern that I find in my DNA may be very different from the cleavage pattern that's found in your DNA. And so we can take samples from each one of us and figure out which one belongs to which person by simply looking at this pattern of restriction endonuclease cleavage. Now, the fragments that are produced by restriction endonucleases in this case are what we call restriction fragment length polymorphisms, or RFLPs for short. Now, we can use a complementary DNA probe then to a particular area of interest, and we can identify particular alleles possessed by a given individual based on these RFLPs. So here's an example. Let's say we have 3 different alleles with 3 different restriction endonuclease patterns in a population. So allele 1 has 2 restriction sites that are quite a distance from each other and produce a long fragment. Allele 2 has restriction sites that are a little bit closer to each other, producing a medium-sized fragment. And in allele 3, we have the 2 restriction sites closer to each other, producing a small fragment. Now remember, each individual in the population will have 2 of these 3 alleles. For example, if you do southern blots on 3 different individuals, you may find that one individual has a long allele that doesn't run as far in electrophoretic gel, and it has the medium-sized allele, or allele 2, that's shown as individual 1. Individual 2 has the long version and the short version and produces the pattern that's shown. Individual 3 has the medium-sized version and the short version, that is alleles 2 and 3. And so you can see that these 3 individuals can be distinguished from each other then using these specific polymorphisms. Now we can use this then in the diagnosis of individuals. We can use it in differentiating individuals from one from another. We use it in forensics to identify individuals who have died and are unidentified. We use it in paternity testing and lots of different things. Here's an example of how we might use it in the diagnosis of a disease. Let's say a couple has a child who has Hurler syndrome, for example. The mother's pregnant with a second child, and she wants to determine whether this child will have Hurler syndrome as well. And so Southern blots of the parents and both of the children are performed using a probe from the alpha-L iduronidase gene. This is shown on the next slide. Now, Southern blotting of this gene reveals that in the father, he has a long fragment and a medium-sized fragment. The mother has a long fragment and a short fragment. And the first child, the child who was affected, has only a long fragment. What that means is that that child is homozygous for that particular restriction fragment length polymorphism. Now, the child for which the amniocentesis was performed then has a genotype that looks very much like the mother. He has one of the long alleles and one of the short alleles. So the question is, will child number 2 be A, homozygous for the mutated allele, in which case he would be affected with Hurler syndrome, B, heterozygous for the mutated allele, in which he would be a silent carrier, or C, homozygous for the wild type or unaffected allele, in which he would have no disease either, or D, none of the above. The correct answer is B. This child will be heterozygous for the mutated allele. Now, the top band represents the mutated or disease-causing allele, that longer fragment. We know that this is the case because the affected child has 2 copies of it. Remember that Hurler syndrome is an autosomal recessive disease. That child must have inherited the mutated allele, one from each parent. Now, child number 2 has a copy of this mutated allele, but the second allele is the short allele inherited from his mother. And so we know that he carries one allele with the affected gene, but the other allele is normal. And so we can predict then that this particular child will not be affected by the disease, but has the possibility of passing it on to his offspring. Now, one other technique that helps us analyze DNA is what we call PCR. PCR stands for polymerase chain reaction. And this is a technique whereby we can take a given segment of DNA and replicate it or amplify it many-fold. This technique is important in amplifying a DNA segment that you want to study or analyze. It's important for identifying the presence or absence of some DNA in a sample. We use this a lot for trying to detect viruses, for example, in clinical samples. And in addition, we can use PCR to quantify the amount of a particular DNA in a sample. This is how we do HIV viral loads, for example. A PCR is a 3-step process. The first thing we do is we heat the DNA to about 95 degrees Celsius. This denatures the double helix. Then we add specific primers. These are short complementary segments of DNA, which are allowed to then anneal to the DNA at a lower temperature, about 55 to 65 degrees. We then raise the temperature to 72 degrees, at which point there's a thermostable DNA polymerase which is activated and replicates the DNA from those primers. So here's an example of how that works. In step 1 to step 2, we heat the DNA, and that separates the strands. In step 3, we add the specific complementary primers, which bind either side of the segment of interest on one of the 2 strands. And then in step 4, we extend that using a DNA polymerase. And you can see that now we've produced fragments which span that region of interest. Now, if we repeat this cycle over and over and over again, what we end up with is 2 to the 30th, or approximately 10 to the 9th, or 1 billion copies of that specific DNA segment, as is shown in this particular schematic diagram. So that's how we use PCR, and again, we can use that for a variety of different purposes. The last technique that we'll talk about is recombinant DNA. In recombinant DNA, we use a couple of different enzymes. One is a restriction endonuclease, the other is a DNA ligase. And this allows us to cut and paste segments of DNA from one location, paste them into another location. Using this technique, we can take genes or other DNA segments of interest and insert them into plasmids, which can be transferred into bacteria for the production of large amounts of DNA as bacteria replicate, or for the expression of eukaryotic proteins in bacteria. This is how this works, and this is schematized here. We take a plasmid. This is a piece of circular DNA that can live in a bacteria. We cut it with 2 restriction enzymes, restriction enzyme that cuts at site A, and a restriction enzyme that cuts at site B. Notice that these are restriction enzymes which produce overhangs. Then we take a fragment of DNA that we're interested in and cut it with those same restriction enzymes. And we use a ligase to then ligate that segment of DNA into the bacterial plasmid. What we do then is we can transfer that plasmid into bacteria, and it can replicate over and over and over again. Or we can stimulate it with certain materials which will cause that plasmid to be transcribed and lead to the transcription and translation of that gene to produce a lot of protein from the bacteria outside of the body. This concludes the section on tools of